Hi everyone, today we're going to be talking about the lineage of leukocytes, which are the white blood cells involved in the immune responses. So we start off with stem cells, in particular one of the most well-known examples of adult stem cells, which are hematopoietic stem cells found in the bone marrow. So these give off two lineages, the myeloid and the lymphoid precursors. So starting with the myeloid lineage, there are a lot of things that can be formed from these, but one of the major groups is granulocytes. So named because they have a granular cytoplasm. So that includes the eosinophils, which typically have a bilobed nucleus, and they are named eosinophils because the acidic dye eosin stains their cytoplasmic granules pink, and they are used for defense against parasitic infection in particular. And another example is the basophil, which also has quite coarse cytoplasmic granules. It can be bilobed and even trilobed, although it can be difficult to see the exact shape of the nucleus because it's often obscured with the granules in histology. So it's called basophil because it's stained by basic dyes, and it's actually stained blue. So that's kind of easy to remember. B for blue, B for basophil, given that you're staining with something like hematoxylin and eosin. And the third example are neutrophils, which don't particularly stain with acidic or basic dyes. They do have cytoplasmic granules as well, though, and their nucleus is can be multilobed. And this is particularly because it's especially involved in the phagocytic response, so it has to be able to undergo sort of diapedesis, which means squeezing out from between the gaps in the endothelia of blood vessels, and engulfing pathogens. Engulfing pathogens. So now we move on to another subset of myeloid cells, which are the megakaryocyte erythrocyte common precursors. So as the name suggests, whenever you have a cell that can give rise to two things, it has to be a precursor because it has to be able to specialize further. This entire diagram is about how a multipotent cell, which could become anything, undergoes this basically decision tree to specialize into one type of cell at the end. So every time you see precursor, think it could be multiple things at the beginning. So this is very clear, tells us it can be either a megakaryocyte, which is a very big sort of immune cell that doesn't last very long like this before it becomes fragmented into what we all know and love as platelets, very important in the clotting pathway. So that's to respond to blood vessel rupture following skin injury or any other type of sort of injury that requires a clot formation, and it's involved in creating a platelet plug around this area. Okay, I'll not sketch that. And so that's megakaryocyte. Mega kind of tells you it's big in size, and erythrocytes, obviously these are the one type of cell on this diagram that are not immune cells, and they specialize for transporting, binding and transporting oxygen. They lose their nucleus and ribosomes early on in specialization. And finally, we have this unknown precursor first, before we get a mast cell. These are responsible for histamine secretion, for inflammation, and they signal and work together with the next type I'm going to discuss, which are actually macrophages. And macrophages and dendritic cells come from a common precursor called monocytes. Monocyte because they have one large central nucleus. Then they split into, as I mentioned, macrophages and dendritic cells, which are the only type of immune cell not to have an effector function, but rather they relay messages. So the way I remember it is, well, two ways really. Dendrites I used to associate only with neurons. So dendritic cells are like the brains of the operation, and also they've got all these dendritic processes, which is probably the reason why they're called dendritic cells, but it's just important to remember these are not anything to do with neurons. They are immune cells, leukocytes. So macrophages are the other type of phagocyte, main type. So unlike neutrophils, which have a very short lifespan and they turn into pus and they die very soon after performing their function, these stick around a bit longer and they can become antigen presenting cells. So they can sort of provide a sample of this bacteria's broken down proteins and display it here using their their MHC, major histocompatibility complex, and this helps to trigger some of the other cells that we'll come to with the lymphoid system. So macrophages also secrete a number of things, including arachidonic acids. So they're responsible for the conversion of, for example, formation of prostaglandins, and these are from phospholipids in the membrane. So I can just represent it like this, meaning this is its hydrophobic tail, and this is its, this is its hydrophobic tails. And this is its hydrophilic, negatively charged ionic phosphate head. So this can be converted by cyclooxygenase into prostaglandins, including prostacyclin, for example, that's involved in pain. And so this is prostacyclin, but there are others like PGE, PGD. So I'm just going to write them in this order and I'll explain why in a second. So PGD, PGE, and PGD, PGI, and PGE, because I remember it as dilatatory. So they're responsible for vasodilatation, among other things. But they're also involved in the pain response in inflammation. So this increases inflammation also. And there's also another thing that uses this same enzyme to form thromboxane A2, which kind of does the opposite, but that's from the platelets. Sorry, not from the platelets, that's from the platelets. And these two kind of act against each other. And that is exploited by some of the antihypertensives, actually, because they can inhibit this enzyme, and because platelets don't have nuclei, they can't protein synthesize more of it, whereas these ones can. But moving back to the other types of arachidonic acid derivatives, you get lipooxygenase. So if this is the fast response, the slightly longer term, slower response is lipooxygenase, which gives leukotrienes. And leukotrienes, so that's also pro-inflammatory as well, though. The other thing that uses lipooxygenase, I would have put it on the other side, but there's no space here, but this also applies to this. Lipooxygenase is used in an even slower, not even slower, but it's sort of a negative feedback when this becomes too pro-inflammatory. It switches to becoming anti-inflammatory, producing lipoxin. And there is a mediator switch that actually flicks it from this onto doing this. So there is a lipid mediator class switch here. So that's one major function of macrophages besides phagocytosis. And this is how they work with mast cells as well. So the mast cells often recruit them. And the other acute phase proteins that macrophages can release are, for example, fibrinogen. And that kicks off, again, as it says ogen, it means it's a zymogen, it's inactive, and it can form fibrin when it's cleaved, which is involved also in the platelet plugs for clotting. And defensin, well, actually that's not an acute phase, but what is acute phase is C-reactive protein, which is involved in opsonization and the complement system. I will discuss the complement system in another video, but I always think of it as like tiny bombs in your blood. And Kurt Gesagt has a very nice video on it as well. So I do not copyright that idea. Full credit to them for that analogy. So, the third thing that, again, another thing, we're not done, that macrophages secrete are defensins. And these neutralize toxins that are, sorry, that's very rubbish skull and crossbones, and they can disrupt bacterial cell membranes. And lastly, pentatraxins. Pentatraxins can bind pathogens sort of like antibodies, because they have receptors that are complementary to the antigens on the surface of the pathogens in a similar way to antibodies. But what they do afterwards is slightly different. So, okay, if I draw it like that, it looks a bit too much like an antibody, so I will draw it like this instead. Which doesn't look much more different anyway, but for an example, serum amyloid protein and PTX3. And the sorts of pathogens that they can bind are not pathogens, but the sorts of antigens they can bind are CD8, 9, and IgE. So that's a lot of the key things that are secreted by macrophages. Moving swiftly on to the so far undiscussed part of our lineage diagram, the lymphoid heritage. So these are the B and T lymphocytes that are commonly taught at school. But before we can get to the T lymphocytes, we have to go through the natural killer cells. And because they are NKC and then T cell, they must be precursors because they are bipotential. They can become multiple things, two things in this case. And I've run out of space, so I will have to go downwards. Apologies. So, they can give natural killer cells, which are involved in the early response, and then important for starting the specific adaptive immune response that acts based on the type of pathogen rather than just the rapid but non-specific innate immune response. And we can discuss the two types of T lymphocytes. So there's helper T cells, which are also important for moving along the process of adaptive system activation by activating the other types of lymphocytes, and cytotoxic T cells. And lastly, B lymphocyte precursors to give B lymphocytes. Okay, I think that might be everything I want to... Oh, sorry, there is one more thing that I wanted to discuss today, and that was cytokines, which are also, guess what, secreted by, this would be number 5, wouldn't it? I might have to do it down here. Also secreted by macrophages, among other things. So other things can secrete them as well. And specifically, how this can be linked to things like fever and inflammation. So cytokines can take on many forms, but one example would be tumor necrosis factors. Specific example of that, tumor necrosis factor alpha, which goes wrong in sepsis, where it's overexpressed, and that might be due to increased, the long-term implications of prolonged secretion of TNF in infection by the very active macrophages. Maybe I was wrong to say overexpressed. It is justified in that there is an infection that it's not autoimmune or anything like this, but basically there are short-term and long-term consequences of too much tumor necrosis factor. So in the short term, there is, well, obviously it can cause death, but if you don't die from it, it can, or while it's killing you, it can cause a drop in blood pressure due to, as we mentioned up here, how the macrophage response includes things like prostaglandins and other vasodilatory substances, agents that can increase vasodilatation and also vascular permeability. And the second one is to vasodilatation around a site of infection. The reason for that is obvious, to transport more leukocytes to be able to do their thing and fight it. But in the case of vascular permeability, it's actually quite a struggle for... So I'm drawing it like this, but there should really be gaps on the top as well. But if this is our site of infection, as the blood flows through these vessels, the hemodynamic forces on all of the leukocytes inside them are very high. So they're more likely to be pushed along this way, and they need a lot of force to be able to overcome it and then deflect off that flow to come out through these. And this is why vascular permeability, that means kind of increasing the gaps in these by... or just making it easier for them to push through these, is helpful for that. And I'll just circle back to it in one moment, the different stages of how all of these cells actually get out of the blood and into the infected tissue to do their job. So this can lead to loss of blood volume as well sometimes. If you have if you have increased vascular permeability, you might get fluid as well, generally, moving, like plasma fluid, moving into certain areas and causing swelling or edema outside in the extracellular fluid, but in the plasma, so the actual fluid volume of the blood might go down. So it can cause decreased blood volume, and that leads to, in the end, septic shock. So this might be a reason that you die from it, or it might just be a short-term symptom of it. And long-term, you have complications like using up a lot of the clotting proteins, because again, the inflammatory response, the response to infection involves, we've seen a few key themes be repeated, and that would be things like the clotting pathway to seal up entry routes, things like dilatation and vascular permeability to increase the recruitment of blood, of leukocytes, of white blood cells, and also things like inflammation. which kind of is a result of all of those. So, as we see here, with the clotting example, you actually use up more of your clotting proteins that can then not be used in other injuries. So you get decreased clotting elsewhere. You also get, because of this low blood volume, if it isn't bad enough to cause septic shock, and or maybe even if it is bad enough to cause, in fact, this is actually part of what contributes to septic shock, you have decreased perfusion. Perfusion is the flow of blood through key organs, and loss of perfusion of these organs can cause organ failure, because obviously you aren't able to transport the oxygen and glucose needed for respiration and for other metabolic processes. So that's also bad. And I just forgot to mention also one other short-term factor that I've just remembered is this. So because you have this increased clotting that we mentioned here at the actual site of infection, so this is elsewhere, and this is in the region that you are in, that you're fighting the infection, you have increased clotting leading to disseminated intravascular coagulation, which is in the small vessels. You have increased clotting. So if you have a blood vessel close to the site of injury and you have white blood cells leaving it, and that's actually effective, that's a good thing. But if you have smaller vessels nearby that aren't as helpful in the response, but they're still getting blood clots, you might get loads of small blood clots kind of dotting around the circulation, and if those travel away from the infected region and break off and maybe go to places like your brain or your heart, on second thoughts I'm not going to try and draw it, then it will cause things like stroke and myocardial infarction if they are large enough. And anyway, they can just disrupt flow. So that's everything I wanted to discuss today. Oh, and I forgot the process by which this leaves the blood vessels. So very quickly. So it starts with has to kind of loosely transiently anchor itself to the membrane where it wants to leave. That's called rolling adhesion, because it's still kind of rolling, and it's still, imagine like seaweed at the bottom of the ocean, it's still kind of being pushed in the direction of the flow. So if the flow is going in that way, it gets swept along a little bit like this, but at least it's not moving past that point. Then you get tight adhesion, where it binds with a bit of a higher affinity. Next, you actually start the process of leaving the blood vessel. So if it's anchored here, it leaves by diapedicis, which means it has to squeeze out like this, and that's why those, in particular the phagocytes, but the others as well, have to have lobe nuclei to allow them to change shape. That's called diapedicis. And at this stage there is also a breakdown of the basement membrane under the arteriole, separating it from the extracellular fluid or the tissue. So this is broken down as well with metalloproteases around in the region around it. Oops. And finally, chemotaxis. Chemotaxis, if you think taxis take you places, chemotaxis is the transport of this, but it's not being passively taxied around, it's actually moving towards the site of secretion of some of these chemokines that actually attract it. So it starts to move itself. So you can also think taxi like with the T for attract, so it's attracted by the chemicals, chemotaxis. And these ones in particular are CXCL8 as an example. Great, and I forgot also to mention, diapedesis is the name for the motion of this out, but the whole process, like on a tissue level rather than just for one individual cell, but for the whole vessel, I guess you could call it tissue if it's, yeah. For the whole vessel, it's called extravasation. Vas being vasculature for blood vessel, extra meaning out, going out of the blood vessel. So I hope this video was helpful. I hope it helped you to appreciate how connected a lot of the responses are and how many cells can work with each other and do multiple functions. And we actually have names for these, so we have terms for these. Pleiotropism, sorry, I know I thought the video was over too, but pleiotropic or pleiotropic. I'll just double check, I don't want to say it wrong. I think it's pleiotropic. And that means one cell, many roles. And then synergy versus antagonism. So they can work together, they can have conflicting actions like we saw with the arachidonic acids from making prostaglandins versus the ones making thromboxane from the platelets. So those two worked against each other because thromboxane is actually important in blood clots. So to dissolve blood clots, we make use of this antagonism. But funnily enough, they use the same enzyme. And then so there is convergence of some of their roles as well. So one role, many cells. And I think now that is everything. So I will thank you for listening and let me know if you want me to make any more like this. And let me know if you found it useful. And if I made any mistakes, also please let me know and I'll correct it. Thank you.